Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute.
From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 30th, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 65th episode of Matters Microbial. I honestly didn't believe we would make it this far, so many thanks to Vincent Racaniello, the Podfather, my deeply awesome guests, and most of all, those of you who watch and or listen to these podcasts, truly. As this episode is being recorded, tomorrow is my favorite holiday, Halloween. You can see how much I enjoy this holiday with a plush giant microbe adorned with a glow-in-the-dark skull mask made by Dr. Jennifer Quinn. The students here at the University of Puget Sound enjoy pranks, such as seeming to put my head in a bottle, seen here. I don't know quite what to make of that. This week in Bio 350, my Micronauts were able to explore a tiny bit of bioinformatics, using their 16S ribosomal RNA data to identify bacteria isolated from their reusable water bottles, their water bottle buddies. Single colony PCR was carried out with universal 16S ribosomal RNA primers. A portion of each reaction was run in an agarose gel to determine if a proper sized DNA amplicon was present. The rest of the reaction was cleaned up and sent off for DNA sequencing. We discussed how to evaluate DNA sequencing chromatograms and then used a few simple tools to identify, their water bottle buddies. As I've seen before, certain bacterial taxa are very common in these water bottles because they're biofilm formers and oligotrophic. Here's a word cloud with some of the bacterial taxa I usually see from student-isolated water bottle buddies. You'll notice this organism Chrysobacterium, which is extremely common every time I do this exercise. In any event, as I tell my Micronauts, the organisms could come from the air, the water that they put in the bottles, the student when eating or drinking, etc. And I often wonder about testing some of the isolates for biofilm forming ability. But this time, a new one. Agrobacterium? The bacterium that can cause crown galls in plants? Without genome analysis, I could not be sure, but as you can see via both RDP classifier and BLAST, this isolate certainly seems to be a close relative of Agrobacterium. Now, I hadn't seen that one before, and I'll freeze away the sample in case I would like to return to it later. Several times on this podcast, we have discussed the Archaea. There is no E. coli of the Archaea with which we can dive deeply into the cell biology of this domain, but that will change today. And since many researchers think that the Archaea are our ancestors, and we certainly have some unique things in common, understanding how these organisms live can become very important. One further thing. Throughout my podcast session, my producer David Renata often asks if a given organism is squishy. This isn't silly, it's the whole idea of getting to that perspective. So I was pleased and amazed to see this quotation on today's guest's very interesting laboratory website. So, I am very pleased indeed to introduce the remarkable scientist Alex Bisson to discuss the work that he and his coworkers do studying the mechanobiology of Archaea and why we should all pay very close attention. Welcome to the Quality Quorum for today, Alex. It's great to see you.
Thank you, Martin. It's a pleasure to be here. Thank you for the invitation.
Oh, I think it's wonderful. And I would like to get right to it for a little bit. And it has to do a little bit about David's comment about things being squishy. We tend to look at things from a microbe perspective. And in fact, it's hard to have a microbial perspective, how microbes can detect a surface, how they can touch one another, how they react to that. And this has some reflections the way that our own cells grow together to form tissues and that kind of thing. So could you, because as I went through your work, I can't believe some of the techniques and visuals that you have. They're really quite amazing. So could you talk a little bit to the listeners and viewers about why you chose Archaea and why everyone should care?
Well, that's a great question. And it's a long answer that I'm going to summarize because it goes through my whole career. First of all, I think that I was lucky to have had very different mentors that would teach me very different techniques and work in different projects. Most of the time I worked with bacteria, not Archaea. The first time I heard about Archaea, I was a PhD student. And when I looked those cell shapes, I said, these are probably all artifacts. People do not know how to take pictures properly and forget about them. Years later, I was in the middle of finishing my postdoc on bacterial cell division. And I gathered a lot of very cool single particle tracking techniques, but I started to get frustrated because the cell wall of bacteria is a very complex structure. And it is hard sometimes with so many pieces to tear apart what is regulating cell shape, cell division, or whatever cell processes I was interested in. Then I had this great, other postdoc in the same lab at Gardner Lab at Harvard, Jenna Jung, that was interested in Archaea, and she was studying size regulation. She was more into the systems biology of Archaea. And then I just saw her picture, say this is amazing. And then they do not have a cell wall. They do only have glycoproteins on the outer surface. That's perfect for any microscopist doing single particle tracking, understanding cell shape. More than that, they changed shape and they did. And so, by the way, in a year to end my postdoc, I decided I'm going to completely switch fields. And you can imagine how people thought I was crazy by doing that, but it paid off because they are amazing microorganisms.
So I want to take just a moment for the listeners and viewers. We're used to the three basic shapes of bacteria that folks see. A rod, a sphere, and a spiral. Bacillus, coccus, and spirilla. And we're used to that, and we know some of the cytoskeletal elements. It's so funny. I that's a sensitive subject with me. People don't believe bacteria have a cytoskeleton. Yes, they do, of course. But what's interesting about it is that some of the Archaea have very unusual shapes. And even though you know this and I know this, I'm going to actually put up a photo of Halo quadratum walsbei. And what you're looking at is a square bacterium. A square archaean. See how bad that was? In any event, this square archaean is fascinating because it may be an adaptation to the high salt levels, but it really speaks to what you're saying about unusual shapes. Then we get into some of the other ones. There are some very unusual shapes seen among the Archaea, and then I don't see that level of variety among the bacteria. Am I wrong?
You're right and you're wrong. As everything, we always when we generalize, we are wrong. But bacteria do have all these shapes, or almost all those shapes: squares, cubes, all sorts of shapes. The special thing about Archaea is how short branches, meaning organisms that have very high similarities to one another and didn't diverge so long ago, they can vary in shape. So that's what I call very plastic. They can change very fast, during evolution. Which means that the genetic potential there of it's very high to change shapes very fast in evolution. And that's something that brought me to think about evolutionary cell biology of Archaea, because something has to explain why bacteria or even eukaryotes do not do that as well. You can see eukaryotes doing that, but not as fast as Archaea do.
It's so interesting to me because, and I hope I didn't give any disrespect when I said this, a lot of the Archaea are challenging to grow. And for that reason, most of our deep knowledge happens to be on bacteria that are easier to grow. You're changing some of that. And I also like very much your commentary about the cell wall and that, and, you know, gram positives have a very thick peptidoglycan layer, gram negatives that have a very thin one, and all the different, and then two membranes on top of that. And there's all this kind of variety that we see. But when I think of the Archaea, I often wonder about the ubiquity of how often we see these things. And what I really appreciate in your work is how you picked a couple of things and you picked up this shoe, which is a series of techniques, and you hit over and over again to make them give up their secrets.
Yeah, we torture them until they give up secrets. So
yeah,
that's kind of like, motto in our lab. Like, we shoot lasers, we stress them in all ways we can, and they give up a lot of like hypothesis-free new discoveries, because we have to explain that it's like a bottom-up approach, that you first try to understand things the way we don't expect and go into the mechanism, instead of like having hypotheses about the mechanism and then go after the importance and impact of that specific molecular or cellular like pathway.
One more thing, I'm so sorry, because I want to get into the actual details of what you've done and what you've found. But I just got through teaching my students about L forms of bacteria. And I was really struck by what you said about a lack of a cell wall, when L forms are very strange beasts. And the problem with them, of course, is that L forms aren't, generally speaking, stable, and they have compensating mutations that occur, yada, yada, yada. All things that are alive have the hand of Darwin upon them. I've got it. But that just gave me an interesting echo of what you're saying. So take our listeners and viewers through some really interesting things that you found, and more to the point, how you know, because what struck me about your work are some of the wonderful visuals. And we'll put links as well for people that are just listening. It's so worth your time. Why don't you take the listeners and viewers through some of these really wild and interesting journeys that you've gone on with these? I mean, I'm looking at the figures and I see tissues.
So that's a long ago story when I started to plan my lab, and I said, well, these little guys are very squishy. So that bothers me because I cannot follow them through a lineage, growing, dividing without being deformed. So always started with me wanting to not deform them. And then we try many different organisms because maybe there is a better model than the other. At some point, Teope Rados, who was a staff scientist in my lab for 5 years, now she's a grad student at Brown University, she started squishing them harder, something that I never tried before. And then when she squished them harder just to see what would happen, if you go all the way down, they start making these pancakes and they start kind of feel very deformed. And I look at them and say, yeah, that's not so interesting. We're just torturing them, right? But then Teope was very wise and she let the experiment go longer. And that's the first lesson that we get. Go, even that you're finishing and ending your experiment for whatever reason you have, leave things on the plate a little bit longer. Be curious about what happens if my plate, dries out. What happens to my cells or my colonies? So that's what she did. But then when she looked at the microscope, she saw this weird embryo-like cellularization, looks like carving out these pancakes, and then looks even like worms, when it's cellularizing, moving apart. And it made like what looks like when I start showing people around, because I couldn't believe. I showed a bunch of colleagues working in archaeal cell biology. They've never seen that. Someone told them, "Oh, this looks like an embryo or epithelial tissue." Then I look at, yeah, it does look like an epithelial tissue. Then TOP let it go, and then we see that it spreads out in this monolayer that is exactly like in vitro epithelial cell culture do, right? They adhere to the surface and they spread out. So then it started us to imagine, okay, what are we looking at this? We are looking cells squishing each other. Is an artifact? Is interesting? Is this a selected trait in evolution, in the natural environment? Are they doing this because they have the genetics encoded to do that? And that we started the whole, it's a long journey of 5 years to finish this paper, because TOP is so careful about every single detail in the experiments. And we had to learn a lot of tissue mechanobiology, because that's not my background, that's not TOP's background. That's when we got Olivia, that is a student that joined it in with a physics background. And then she said, I want to start doing proper mechanobiology experiments with them. So TOP on the cellular physiological level and Olivia on the mechanical level joined forces, and then we went all the way with the paper.
So this is an emergent property of different people coming together. And one of the things that I don't know if listeners and viewers appreciate is how collaborative science is, and not simply with different subspecialties within an area, but sometimes the view you get from outside. And this is a wonderful thing to see. May I ask how you squish cells technically?
Oh, yeah. There are very, very easy ways to squish them. For example, you can make the same way you pour a petri dish's agar plate with media for cells to grow. We do that with agarose, with different concentrations of agarose. Mm-hmm. So TOP, when she titrated the agarose on these pads that we call pads, but basically is gel-like media, she started from 0.25% agarose, that is the minimum we could make it, that we could manipulate, and went up to 6%. That is the maximum that we could dissolve in our media. And she tried a number of concentrations. Then she cropped a little circle or square of this plate and just put this pad on top of a droplet of liquid culture against the cover slip or glass. So it's a sandwich. Mm-hmm. And this dish, this kind of sandwich that is rapid to not dry out, goes to the microscope, and we image cells there.
It's amazing.
So it's very cool, because you see that nothing happens at 0.25, 0.5, 1, 1.2. At 2%, all of them change to develop to the tissues. And then doesn't matter the concentration you put more, they keep developing to tissues until you get to too high, 6%. Then they cannot support anymore, they die. So there is a maximum amount of pressure or compression that you can use against them.
So this idea of mechanobiology is a relatively new idea to me, but it makes a lot of sense. Again, taking our perspective down to the microbial cell level, when I say the word perceive, I'm not inferring consciousness. People think that I am, but I'm not. So what are they sensing when these things happen? How do they know they're being squished, for lack of a better term?
Yeah, no, that's a huge still mystery, especially for Archaea. But if you go to microbes, it's very important for them to sense, or at least to detect, when things are crowded. For in our guts, there's a lot of stuff going on there with the microbes. So they squish each other, or when they reach the blood current, and then they're sensing shear. So sometimes, just before going to the answer of the question, just to contextualize, because Mechanobiology is not so popular among microbiologists because the number of ways molecules can signal behavior, it's it goes to endless ways. They can combine with many different things, and people feel that shear and compression and touch, all these things are very limited ways of transmitting information. But that's not true. We humans ourselves, we interpret it with our receptors, many different types of touch and cold and warmth, that is very, very detailed and well resolved. So the question is, do the microbes do that? So, and how to do that? That's your question, right? So there are many ways that they could do that. For example, if you're talking about bacteria, they could be sensing through their structures like pili and flagella, and depending on the shear and then the compression, they could transmit mechanical information and transform this in biochemical output. Archaea itself, we believe that what they are sensing there is when they grow and they stretch their membrane, or because the membrane gets decreases with it, or the membrane gets more sparse and less dense as they stretch, increases the tension. This starts to trigger conformational changes in receptors and membrane proteins that will change the conformation, will interact with the proteins, the cascade of signaling inside the cell fires. And then the genetic expression program changes through that. So that's the interface physics and what we call biology or genetics. We have to remember that everything in the cell is a physical thing, just like we are. They can be touched, they can be stretched, they can be compressed. And the enzymatic activity in the cells change depending on these factors.
Do you know the story in Vibrio parahemolyticus of how they sense a surface?
Not specifically about this Vibrio species.
So the whole thing is, and this is work from Robert Belis and Linda McCarter many years ago, but the polar flagellum acts as a dynamometer, and anything that impedes its free rotation, somehow that information is signaled and changed into gene expression, those lateral flagella they use for swarming motility. And that's what got me thinking a little bit about your work. I mean, how is it that you're sensing things? And I often tell my students that we have prejudicial ways of looking at microbes. We grow them in culture tubes. That's not how they grow in nature. We grow them by themselves. That's not how they grow in nature. We grow them on petri dishes. That's not how they grow in nature. And so what I find amazing is that you're describing things that are taking place in nature that people haven't looked at yet.
Yeah, neither we did, right? I'm overly excited to start adventure myself into the Great Salt Lake in Utah and start looking for these tissues, because we can reproduce many conditions and many variables from the environment, but we will never be able to reproduce all of them or most of them.
Right.
So as a lab rat, as myself, I think I've never done field. I'm not a biologist, originally. I've never done field work. So I imagine, now I have to learn how to do that, because that will be very interesting to see how cell shapes and behavior happens in their natural habitats. But yeah, I feel that we do not comprehend, even for E. coli, Bacillus thurilus, staph, what they're actually doing, where they are doing. It's very different. I call it the lab a minor league, the baseball minor league, and then the nature doesn't care. My major league doesn't care most of the times about the minor league, right? Nature is doing their own thing.
Do you know how many years it took to get the term biofilm put into introductory biology textbooks? It took years and years. So these paradigm shifts are a big deal. And that's what I really appreciate about your work, to repeat myself, is you're talking about actually looking about how organisms are interacting in ways I'm not saying they're the same as in nature, but they're closer to nature than what we're actually doing in the lab.
Yeah, can you imagine what archaea are doing in the guts, in the human guts, or
Yes,
methanogenic archaea are doing in cows, for example? It is a whole new, and let me clarify. Let me clarify because I'm very proud of our work, but the archaea field. You earlier mentioned that archaea are very different to cultivate, and that's why we are behind. I think it's much more, in my opinion, maybe other people have different thoughts. Archaea are not like a little behind other microbes in terms of number of labs studying, number of papers, because they are hard to cultivate. Most bacteria are very hard to cultivate as well.
hmm
I think it is historical and cultural. So I think that we were not paying attention to archaea, maybe because they're not pathogenic, or we haven't
sure.
found one that is pathogenic, or maybe it's just that it took time, right? Like for people to start looking at them. And then that's when I have to think, like, the field of archaeal cell biologists or biochemists or everybody in the past 20, 30, 40 years have been working so hard. So someone that is crazy that can just say, okay, I'm going to get a fast track training in Archaea for a year and start my lab. They allow it, like, they paved the way so my lab could do that. They took me into their labs, they trained me, they share all their resources. It's a great community to be part of. There's no way of doing new science or moving fields without community so when you say that my lab is doing all that, I think that our community is doing all that, and just a piece of the puzzle there.
And I really appreciate what you're saying. One of the things that's always struck me, and maybe I'm the term that I use growing up is Pollyanna, where I tend to look at the more positive, but I have been really, really pleased, especially in recent years, is how collaborative and helpful microbiologists have been toward one another. And sometimes we don't talk enough. And this is a good example of that. And what I appreciate are all these people that went out of their way to bring, if you'll forgive me, you more knowledge in their area, and then you built on that. And at the same time, you show gratitude for it. And these are things that I want listeners and viewers to understand, that science has fundamentally changed the way it used to be in terms of being much more collaborative. And I hate the term welcoming because it sounds like a silly term. I don't mean to diminish it. I think it is important to be welcoming, but that has been my experience in microbiology.
No, that's true. Completely. And then I feel the Archaea community, and then that's my message to all people that are doing whatever science they're doing. Come and join and start working with Archaea. We all gonna build you up with like belts and tools that you need to actually do the work. We need more labs doing Archaea biology. So
And also you were able to use a number of tools that I was going to say that you put into this study, and I understand that you were one of many people that were able to translate tools, for example, commonly used in gram-negative lab rat E. coli, so that you can actually start to do things like gene knockouts or regulatory mutants or knockdowns, things of that nature that enable you to ask and answer really interesting questions.
yeah, that's true. So Helophyrax volcani or volcanii, depending if you're a European or American. But Helophyrax is the E. coli of the Archaea, and the reason for that is that its whole genetics is very yeast-like. Like markers, strategies, pop in, pop out. The way you delete genes, the way you suppress genes is very similar to yeast. So every single genetics aspect that we did in our work come from all other labs that was already built on, you know. I think that the contribution that my lab brings in this paper and other of our papers is the biomechanics and biophysics and imaging techniques,
Yes.
and the ability of growing hundreds of different Halo Archaea, which people can do, but we are the first ones to start actually imaging that level, because we saw there's a huge advantage to learn by comparative genomics, by evo-devo. We can learn what is going on with the processes we are watching when we compare microscopy and genomics.
Also want to bring something up to listeners and viewers that may not be as conversant with the Archaea as they could. Something you mentioned before we started recording is something you do with your students where you bring in Himalayan pink salt and you're able to recover some of these halo-tolerant Archaea from them, you said about 30% of the time. And I have to tell you, everybody has these, like, I actually have some halite shot glasses that have pink coloration to them. And Alex, I put one through the dishwasher. That wasn't very smart, but that's just me because it dissolved. But the point is that we've all seen it. And we've all seen that red coloration, and that's part of many of the Archaea that you're talking about. So that really isn't something unusual or strange. It's something we have seen, and folks like you have started looking at and analyzing.
Yeah, like actually, it is an interesting question that people have been thinking for decades right now. Where does the pink color comes from? And then some people, there's not there, as far as I'm concerned, I couldn't find one source that actually shows, yeah, the pink salt lakes are pink, or the Himalayan pink salt are pink because of Archaea, or because of rhodopsin, or because of carotenoids. There's no direct evidence, because salt lakes also have red algae, brown algae. They have all sorts of microorganisms there. So this is up to be investigated. It's a ecology of microorganisms contributing. It's only one, but in the lab we know that when the field deletes, we just use the strain that was generated in the field. But when we delete the gene, the first gene of the pathway to synthesis of carotenoids, there is this pigments that go to the membrane of Haloarchaea and many other bacteria as well. They are not pink anymore. They're white. So rhodopsin by itself, it's not the cause of the pinkness. Maybe carotenoids and rhodopsins are interacting and they have this, but the pigmentation comes from carotenoids. The same way why your carrots are orange.
You know, it's interesting that you bring this up, and I want to thank you for what you just said, because over and over again, we will hear observations as fact. And then you find out when you dig into it, just as you described, that there might not be evidence for that. It was just something that people thought was correct. Very similar to what I still hear people say, there are 10 microorganisms per human cell in us. Turns out that that number was 100% an estimate that someone made that other people started quoting. The difference here is that now you have evidence about things. You actually did the experiment.
Yeah, definitely. And that's the advantage of being new in a field. I go to that, or like my lab goes, and then we have this child's, point of view of, oh, that's very cool. How do you know that?
Yes.
So I keep asking my colleagues, like, oh, how do we know that? Can you send me papers about this? And the papers from the 60s, 70s, 80s, 90s are beautiful. But sometimes you cannot track back that claim, for example, Archaea is squishy. We didn't have a very good piece of evidence in literature, but people talk a lot. And then Olivia, in our paper, did the DMN experiment that is basically squishing the pads that we're talking about, see how much they resist and fight back. And then we see how much of this energy or force is being transmitted to cells. And then we can now say with this graph, this is one graph that took Olivia's 6 months to generate, but a lot of work to learn how to do it, but it's beautiful. Because with this one graph, now we can say Archaea, or at least Haloarchaea, or at least Halopharynx volcanii, is as squishy as mammalian cells in vitro or in a petri dish.
And that is just wonderful. I want to change gears a little bit. So when you, please forgive how this sounds, when you squish this organism, you get what appears to be tissues forming in response to that. Mm-hmm. And they look observationally similar to tissues that you might see in eukaryotes. Now, this must have been an exciting day when you saw this.
Terrifying as well. Because everything starts with we have this very exciting observation. And we have, on the other hand, our very consolidated science program that we planned to carry on with. And something has to give because you have to invest the resources, even time to write grants. I wrote grants for this project thinking they're going to put me in a madhouse, they're going to, because it is crazy. But then when we commit to something, we're going to go through the end. It was so exciting. Every single week, talking to Teope and Olivia, seeing the results got us more and more excited. I think technically was a great training ground because they learned so much about what works and what doesn't work.
Did you ever hear, and I may be misquoting this, and so if anyone is angry about me misquoting this, my apologies. I heard it ascribed to the late science fiction writer Isaac Asimov that science progresses not, and he's being silly, by a damp Greek emerging from his bath shouting Eureka. But where it happens is when someone looks at something in the lab and says, that's weird. And I cannot emphasize, regardless of who said that first, how true that is. And the look on a particular researcher's face, because they're hopeful, we all like those results. You repeat it a number of times to make sure it's real, sure. Now, do I understand? Please tell me again. I believe at least one of these two students that you mentioned that did this work were undergraduates.
No, Teope was already. Yeah, she was a research staff.
Right.
She didn't have a PhD. She's pursuing now, but she had a master's already. And Olivia is a PhD student candidate right now.
Wonderful. So these are people that are relatively early in their careers, and they had a question, and they had the right kind of person around, that is to say you, that gave them the freedom to pursue it. Because there's a lot of energy invested in all this. And look at the payoff. So you have a number of truly beautiful images and videos. And I wonder if you might be able to share a little bit about what they mean and how you did them. I mean, I will have links to all these things that people can look at later, but some of them were just beautiful physically to look at.
Yeah, no, they are. And so some of my favorites are, for example, when Halopharynx volcanii switch shape. So not only tissue is a developmental program in these guys, they have multiples. Just like Caulobacter would also develop into two different cell types, and Bacillus can sporulate, or it can make biofilms. It can make chains or be swimming. So it's not new that microbes are very good at developing to different groups. So one of my favorite videos is to show Halopharynx can switch back and forth between rods that are motile, they swim, they have an arcella. They are arcella, not flagella, but the different proteins, but they serve it the same way to push, in cells ahead. They can switch to discs that look like polygonal flatter cells that instead of arcella, they have pili. that grabs, and then these are the biofilm producers. So you have a colonizer and a pilgrim, like, prospecting ground.
Right.
And then you earlier mentioned it about Haloquadratum being like square and flat. I think this is happening the same way with Halopharynx when they switch between rods and discs. The aspect ratio changes. Discs are thinner, just like Haloquadratum looks like a cracker, like a biscuit.
It does.
They increase the maximum they can the surface area, and that might be important for buoyance, for many things that we still do not know. But you see that the physics and the shape changes and the behavior changes. So this is one of my favorite videos, them swimming or staying put. We had a paper this year talking about how bactophilins, that we call halophilins because they're in Haloarchaea, help to secure the cell shape or the cells to be like, in one piece as they twist and change their shape. So there's a lot of mechanics going on. You wanted to rebuild your tent when you're camping. It's better for you to make sure that you're not going to bend any, poles or anything that you're going to break. You have to engineer things to go fast from one side to another. But for this paper, I really love also the laser ablation. I think one of my favorite movies, the laser ablation, because it shows that was one of the things we want to do. Well, what do plant and animal tissue labs do to study the biomechanics? Laser ablation is one of them. Let's study the elasticity of these tissues. And what we found is that, cells all compress against each other. They do not change their elasticity and tension compared to cells in liquid. But tissues have a completely different biophysical profile or material profile comparable to animal tissues, for example. So it's so cool to see you ripping that apart and see how fast this membranes retract. And this gives us like a lot of information. What proteins might be there, what is different in the membrane that it's not in the unicellular developmental program.
So these are fairly complicated machines that you're using to determine these characteristics.
Complicated is always subjective and relative, right? Honestly, most of our experiments, they are done with microscopes that are in most facilities across the country. We didn't use any specific ablation laser. We actually adapted a purple laser with a very, very that is used for FRAP, but that is important. But the only thing that we need is a high energy and be able to focus in a very, very small volume. But we didn't have any specific equipment for any of that. We had some microscopes that we have in our laboratory. Sometimes we borrow from other people. We adapt as we go, depending on the question.
It's interesting because I could imagine someone hearing this and looking at the images and saying, well, this must take, please forgive how I put this, $250,000 pieces of equipment. But what's interesting is that when I talk to you about it, you say, well, that's not what we did. We have this other way of doing it. And if they hadn't brought that up to you, those people would still be walking around thinking it would be really hard to do that work. So you just have to proselytize that we can solve these kinds of problems. Now, I'm not a physicist, obviously. And so the idea of the biophysics of this may be very, very obvious to biophysicists. But not to me. So I would have said, oh, there's no way that I could do things like this. I'm not saying I should. I'm just saying that's something that I could say. But I would be wrong in saying that. And that's important for people to understand. What is the question and how do you get there? Sometimes I think we get involved with the machinery too much.
Yeah, I think that it is always a balance between the scale of the spatial scale, the time scale. What do you want? Each question depends on different techniques. If you are always like going for the molecules, there are too many molecules to pin down. Like we did RNA-seq to find for the genes responsible for each process. But as history told us, it takes decades to figure out each pathway, each mechanism. It relies on many labs, not only one in 5 years. And as any assistant professors, we don't know if we're going to be here in 7 years, right? So right. We want to get the story out. So that's why we split between physics and molecular biology, because the physics can tell us a lot without knowing exactly which molecules are doing, at least for the behavior.
So I just wanted people to know that you've done a couple of experiments that I read about where you looked at the tissue form and the modal forms and asked about differences in gene expression. And that's a technique that used to be thought of as being very difficult, but now it's something that's fairly easy to, is more straightforward to do and answers certain questions as well. I mean, I'll give you an example. I'm old now. It used to be people argued about it, but now no one does, which is okay. So the idea of being able to get a whole genome from an organism, take that organism, take it through some kind of evolutionary selection in the lab, and then seeing what genes have changed, I do that with undergraduates. And I never would have guessed that. So once again, it's the issue of how the field changes and things become more accessible to other people. The joke that I make is this will be a sequencer in just a couple of years.
That's true.
Yeah,
that's true. I think that the secret now is academia got used to like shape it up or training people to be experts and very good and a few things, and they push it science ahead, and there's no problem with this model. However, now I think that there is a higher diversity of people who learn from each other's and say, okay, I've never done RNA-seq before I started my lab, ever. Before people say, don't, it takes too long. And now I just said, well, Sebastian Kadener here at Brandeis is doing in flies. Why we don't learn with them? Or Vikram Alva and Pedro Escudero, who are collaborators in the paper, are great in, with data sets that are large and complicated. Let's learn with them. So I think that the idea that you can be more polyvalent and then, diverse in your skills, I feel that this is a very important, basically how to solve problems is the best thing a scientist can learn. Things that most students, most postdocs are not going to end up having their own laboratory. They have to be smart in solving problems in general. And maybe even as a PI, you're going to have to be good at solving problems as well. They are not in your alley anymore. So
no, I mean, it is really an amazing world. I mean, the microbial sciences are fascinating. So and the imaging you do, as I've mentioned before, you have some beautiful images of different parts of what's going on in a cell. One of the figures that really struck me when I was looking over things, Alex, is this morphological plasticity of the halo Archaea. Would you mind telling me a little bit about that figure?
Of course. It's an amazing story. When I started my lab, I had this idea that looking at how shape changes would be very hard just by looking at genes or SNPs in genomes, mutations, especially because it's not my expertise, but from experts, it's already very hard to do comparative genomics to see what is circumstantial and what is causal, in all this. And more important, probably many things contribute to a phenotype. So by working with some few species, I start to see, they are very similar. They're over 92% similar in genome sequence, identical. So other things are contributing the system for these morphological differences. And I thought about this idea about, the physical properties, elasticity of cells that can be used to build a phylogenetic tree that is not made out of genes, but is made out of material properties. But to do that, you need a lot of them. So I reached out to Viratiel, that is a curator, it's a PI in Japan and a curator of the German strain bank. And then we established a collaboration where DSMZ sent to us all the Archaea collection, over 100 different species that were never imaged. They were there for years and years. Someone did a very good work of isolating, characterizing, sequencing the genome, giving a name, and then it was there, frozen or desiccated in their collection. And then we are microscopists. And then what I said, I want to image every single one of them. I call this the Archaea Atlas, that or Atlas of morphological Archaea cells. And then we start imaging, and we have imaged most of them by now by like using different techniques. And we found a number of amazing. For example, everybody loves Loki's tentacles, going around. And then you can see there in that panel that there is a Archaean with tentacles. And one of our postdocs is showing that they are membrane bound, they retract, and they advance. So maybe we can learn some biomechanics and biochemistry that can help us to understand Loki a little bit, at least in terms of behavior.
That's amazing. That's what I was wondering about, because as I mentioned before, making a note to myself, this business of the Loki Archaeota are so interesting to me because of this tentacular aspect, with the idea that they might be possible to bring in large food items, and that might be how we kind of happened. That's the whole story behind that. But this is wonderful work. Again, and you must understand, you and the listeners and viewers, my eyes are really bad, so I cannot do good microscopy. So I really adore seeing things clearly in images that you've presented, because I don't see stuff very well through a microscope lens anymore. So thank you for doing that work.
Oh, thank you so much. Really appreciate. And now we put a lot of time and love in our microscopy. That's our favorite, tool and then learning how to do proper microscopy is half science and half art. It's kind of, I always say that it's like cooking. You have to develop intuitions and what is wrong and how to correct, even though you need a solid, conceptual background to understand what is going on. So,
it really is almost like art when you think about prepping samples, things of that nature. And when I say that, it used to be that scientists would kind of give me the side eye, which I understand. That happens to me a fair amount. But what's important is, to some degree, it is art. And that's how the early days of microbiology were. So maybe things have come full circle in a way. I hate telling students how many electron micrographs were taken to give them the one good image that they see published. I hate telling them that, because you can just see the crestfallen look on their faces. No, mitochondria don't really look like that, that sort of thing, right?
Oh, there's a very cool story that I always tell my students. When I was an undergrad in a lab, and then I got excited about biochemistry and everything, my advisor came to me and said, I haven't accepted you yet. You have to go and then get this protein concentration assay and build a standard curve that the R square is higher than 0.9 or 0.99. Hit a very high number. And I said, okay, then I'm going to be accepted in the lab. And then I tried for a month and I couldn't. And then when I showed him the results and then I couldn't understand why, he started laughing and say, these reagents are always spoiled. Nobody will ever get to this, but I like it how much you try it and how interested you are. And then I just said, and then I still excited about the lab. So he accepted me. And then it was a very cool experience. That's when I decided to be a microbiologist, actually, because it was not a microbiology lab. I worked with schistosoma and I worked with rabbits and mice. And I decided, one, I don't want to kill this poor furry things. Two, I have so many ideas that I want to test fast and hypotheses I want to test. Working with higher organisms takes so long. So I love it to move. When I learned how it is to work in a lab, I wanted to move in a bacterial lab or a microorganism lab
Yeah.
that I could do one experiment per week and analyze the data.
That's important. And I'm not casting aspersions on any other field. What you're describing is something that I felt. I mean, when I worked on Cynorhizobium in graduate school, it grew more slowly than E. coli, but that was all right. I had friends that worked on Mycobacterium that took a long time to form colonies, if they formed colonies. So you've got to find a system that works for you. And it's the questions that are important to me. Anyway.
Oh, yeah. Many people have one or two questions and they are obsessed about them. And they will answer those questions in 5 years. And they are happy about that. And I think that, so I also always say there are many ways to be a successful scientist and do science. So that's the beauty of it. And that's why we are good at different things and then we can collaborate.
So Alex, there are so many videos that you've made available that look wonderful, that are fun to share. And I think that you have one that's showing the effects of squishing on some of these bacteria. Can you walk us through that one?
Oh yeah, definitely. So you can have two different classes mainly of squished archaea or species that you can squish. One, as we've been talking, that is, for example, Helophax volcani. It grows like a pancake and then carve out, but does not separate and forms tissues. And the second one is the species that do not develop to tissues. In this movie, you're going to see Helobacterium salinarum that is squished. It pancakes out, but you see that it tears apart and it divides into these polygonal shapes. It doesn't like that much to be squished, but does not form tissues.
Alex, I'm really interested in these variations you see in shape during various stressor events. And are these, I hate the word artifact, are they a consequence of the stress, or is there programming that appears to be taking place?
Yeah. For instance, when we see Helophax switching back and forth between discs and rods, it's pretty clear and established that it's a developmental process. There are different cell types and it's selected. And then we can see that there are Archaea that do not switch from one to another. So they do not have the genes or the regulation necessary for that. In terms of tissues, it's a little bit more up in the air, but our data suggests heavily that it is a selected trait, given the RNA-seq and polarization and the defects that we see in tissues that when we delete certain genes, when we do not see in liquid culture, when they are unicellular. So still, we need more work to prove how exactly this is being selected and what is regulating, but has all the evidence that points that it is also selected and developed.
That's amazing. I have a question about the process of squishing, Alex. I know, how many times has the word squishing appeared in this podcast? It's great. So when you're doing that, are both sides, I hate the term sides, but you know what I mean, are they both accessible to nutrient media? Because it made me wonder whether there's a change in surface area. I hate to trivialize it, to take out the garbage and bring in groceries.
Yeah, definitely. So the answer is the only difference between under the pad and on top of the pad is oxygen viability. And we do see this in RNA-seq, that anaerobic pathways light up under, but not on top of the pad. However, all metabolic, protein synthesis, RNA transcription, and so on, all these processes are not only active or in the same level, some of them are upregulated, and they grow as fast as the cells on top of the pad. So
that's crazy.
So they do the media that is inside the pad is more than enough for them to chew up and then go on.
That's wonderful to think about. I mean, when you think about covering things up, you wonder about stuff like that. But when I sounding trivial, when I streak bacteria on plates, that surface is not dry. We think it's dry, but it isn't from a bacterial perspective. And so that's what made me think about that sort of thing.
Yeah. So one swimming pool is the other ocean, right? Like, so.
Right. Or a slip and slide. Those are my two choices. So something that we started to talk about and didn't finish is the relationship between, and I'm just going to call it tissue formation among these archaea, and the evo-devo aspect of development in eukaryotes. And I think that would be a nice place to start wrapping up the podcast.
Mm-hmm. Yeah, that's a tantalizing way of seeing things. How you in evo-devo and you see the tracks where archaea are making tissues and not making tissues. Do Asgard archaea make tissues? And I think it's very exciting, but might be a little bit anticlimax to hear that. The archaeal tissues that we found have nothing to do with an animal or plant tissues or any tissues at all. This shows the beauty of how nature doesn't care about how we think about systems, how they evolve, how they change, how they adapt. So nature is always recreating and repurposing new molecules and new pieces to get to similar outcomes that probably have nothing against. I think that in evolution, what tells us is that the force distributions and viscoelastic or material properties of tissues is probably a very optimal solution to be reinvented 2 times. And the second conclusion is that multicellular complexity or complex multicellularity, it's not such of like an unreachable outcome as we thought before. Maybe there are many pieces that can be moving, but maybe we are not probing nature and diversity as much as we should to actually get to this kind of conclusions, to minimize saying what is possible and what is not with the pieces we have. So there's no actomyosin in archaea, as far as we know. There are no cytoskeletal motors or complexity in the same way. Anyway, they can build tissues that look like animal and plant tissues. So I feel that the evo-devo was very important, not only to as a starting point to start comparing these tissues, how they look like, they are different or they do not form. And we also found more Sarcina-like multicellular organisms that were not described. Now we have, and one of them, by the way, has cell wall. It's a pseudomurine containing archaean. So is that related to the squishiness, not being squished, having to develop different strategies? We are very excited to use now because the best studied cell-walled archaean is one of our gut. Archaean is anaerobic. So Smithii is very hard to use, even though there are amazing people doing work with it. Now, maybe this is a very good model to study cell wall in archaea because it grows fast, is aerobic. Maybe there's even genetics. That's what we're trying to do right now. So evo-devo gives you a lot of outcome for very little input.
No, this is you have such an impressive laboratory group doing things in directions that you might not predict. One of the things that I keep coming back to again and again with this podcast is how important different faces and different voices and different brains are. And, I can look at that in terms of the way that physicists that came into biology in the 1940s and 50s created modern molecular biology. And I'd be right about that. But I continue to think bringing people in from outside to ask that, and there's an expression from, please forgive me, Zen Buddhism called the beginner's mind. Too many times we think we know the answers to something or something is too simple of a question, when sometimes it's the most important question. So bringing people with different experiences in really matters. I'm going to urge people to really have a careful look at your laboratory group website because there's so many wonderful materials there, so many great images, and it is very clear that you're passionate about making science a good place for everyone and encouraging the spirit of investigation. And it's been wonderful meeting you and chatting with you about those things.
Thank you. And if I can say one last thing,
Of course.
is because of the spirit of my lab, please, if you're interested in studying tissues and want to be a postdoc, students as well, but postdocs, because I want you to take all this stuff out to start your own labs. And then we have a lot of labs that are friends and can collaborate and build up this community. Just one lab is not enough. So if you want to be a postdoc and study mechanobiology of Archaea, please contact us.
That's wonderful. And this is exactly what I've been telling people about the field. Thank you so much for your time. I wish you and yours the very best.
Thank you, Martin. That was fantastic.
Appreciate your time. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Alex Bisson is an assistant professor in the Department of Biology at Brandeis University in Waltham, Massachusetts. Many thanks, as always, to David Renata for superb editing and squishy questions, and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.